So why are we working on rhinos? Okay, all rhino species are endangered. This is where I get my soapbox. They're all endangered due to habitat loss and poaching. So almost all of them have been impacted by humans, and I don't know about everybody else here, but I really feel that we need to do something about this. It's our fault. We need to really help this species. So we're focusing in on the northern white rhino. There's two subspecies of white rhino. Northern only has 2 living individuals left, a mother and daughter, where the southern white rhino has about 18,000. Both of these species have gone through severe genetic bottlenecks. The southern white rhino was able to recover from that. Northern white rhino, more habitat, more poaching. We have really no way of saving them through traditional conservation methods. We need a lot of different things in our toolbox, and I can't emphasize enough how lucky everyone is who works on human and mice. You have all of those tools available for you. We're about 20 years behind you. We do not have a reference genome. We don't have TACMAN probes that we can just order from Thermo Fisher to make our research easy. So as we dove into this project, we're really learning as we go along what we need to figure out to save this species. So the next step, we wanted to make sure that our stem cells could differentiate. So we generated embryonic bodies. We did about 2-week differentiation. So we started with our aggregate culture for 7 days and a plated culture for an additional 7 days. And we do see markers of all 3 germ layers. So we have mesoderm, endoderm, and ectoderm. And I get tears every time I mention this. These are early neurons from Nola. She was the last northern white rhino that we had living at the safari park. So that is now our favorite picture ever. So this is just kind of an overview. Hopefully I've convinced you that we have enough genetic diversity in our frozen zoo in order to save the northern white rhino. We're pretty successful in reprogramming those fibroblasts into induced pluripotent stem cells. It's a very long and tedious process. We have a lot lower success rate than those working on humans do. And it takes about 3 months for us to get to a stable cell line point. Hopefully I've convinced you that we've successfully validated these iPSCs. We've used several different methods of quantifying these cell lines, and we're pretty confident in all of our data. We have our primordial germ cells. We have spontaneously generated ones, and we're also pretty confident that we're generating them with the directed differentiation protocol. Again, this is going to take a lot more work. There's a lot of very small steps in this process, and we're still at the very beginning of this whole program. And then ultimately, we want to get to gametes. We do not know how we are going to do that yet. They still don't know how to do that in humans. Mouse, they do it through a reconstituted ovary where you can do a co-culture, in vitro, develop them. We need to figure out how to actually make those support cells in vitro. They use embryonic somatic cells from mouse in order to do it in mouse. We will never have embryonic somatic cells from rhino in order to do this. So we need to figure out how to do that in vitro as well as make the primordial germ cells in vitro. So there's still a lot to go. Hopefully I gave you a little bit of a flavor of everything that we've been tackling. We've been only working on this part of the project for about the last 3 years. So we've made quite a bit of progress in that short amount of time, and we're diving forward with the remainder.